Hello and welcome to Research Matters podcasts. Today Aishwarya Viswamitra, will give you a summary of science stories that were published during the week on Research Matters. In your weekly dose of Indian science news today, I cover stories on neuroscience, ecology, geography, and cancer. Let's kick off this podcast by talking about dung beetles. Now, don't be repelled. Yes, dung beetles consume animal dung as food, but they are extremely important to the ecosystem. Their unique diet improves the soil structure and soil fertility. Plus, they are instrumental in the dispersal of seeds present in animal species. The earth-boring dung beetles, belonging to the Geotruppidae family, dig deep burrows in the animal dung. In these burrows, the male and female beetles mate, the female lays its eggs, and finally, the eggs hatch. Now, scientists from the Zoological Survey of India, Kolkata, have found a new species of earth-boring dung beetle in the Tawang district of Arunachal Pradesh during one of their field expeditions. Called Enoplotropis tawangensis, it has been named after the district it was discovered in. These beetles are exceptionally beautiful with their splendid natural colors. They have a dark blue body with a slight greenish tinge, blackish antennae, shining blue legs, and light yellow eyes. The male and female beetles look different, with the males having longer bodies and frontal and pronotal horns. In fact, within the males of the species, well-developed males have longer frontal and pronotal horns compared to younger males. This may give larger males an advantage in male-male competition during mating. With the constant threat of global warming and mass extinctions looming over our shoulders, it's easy to forget that most of Earth's biodiversity still remains unknown to us. It's truly beautiful to be able to solve this puzzle one species at a time. Apart from adding to the uniqueness of our planet, many organisms assist us in various ways. A recent study, published in the journal Geophysical Research Letters, used porites, a kind of coral in the Red Sea, to reconstruct the intensity of the South Asian monsoon in the past 250 years. That sounds absurd. How did they do this? The circulation winds in the Indian Ocean are responsible for the South Asian monsoon. These winds also cause dust storms over the Red Sea, bringing the element barium into the water from the Tokar Gap in Sudan. As the corals in the sea grow, the barium in the water gets deposited into their skeleton. The researchers extracted the corals that were known to be over 250 years of age using a hydraulic drill in order to understand the historical variability in Indian monsoon. Just like rings of a tree, the coral cores displayed distinguishable bands that formed every year. The ratio of barium and calcium in these samples represented the amount of dust in the Red Sea. The dust measurements were then tested against Indian sea level pressure records and Northern Arabian Sea surface temperature records, and thus, the barium and calcium concentrations served as a proxy for the wind strength of the South Asian monsoon. The study not only found that the monsoon season has been getting more intense over the previous centuries, it's also the first study to observe monsoon variability over the decades, and they found that the variability of the monsoon has been decreasing over the past decades. Jumping from barium to platinum, cisplatin, a platinum-based chemotherapy drug, has been approved by the FDA for the treatment of a wide range of cancers, including breast cancer and cervical cancer. This is important given that in India alone, 1 woman dies of cervical cancer every 8 minutes, and for every 2 women newly diagnosed with breast cancer, 1 succumbs to it. Cancer is a condition where cells divide rapidly without control, giving rise to a mass of cells called a tumor. These abnormal cells can originate from anywhere in the body. The causes of cancer can vary from genetic mutations to carcinogens like tobacco, and can even be caused by infections from bacteria and viruses. For example, the human papilloma virus or HPV causes cervical cancer. Chemotherapy is a procedure where anti-cancer drugs are administered to kill cancerous cells, and is the most successful treatment method. In a recent study, Dr. Sumit Ghosh from Indian Institute of Technology, Kanpur, discusses cisplatin, the first ever metal-based drug used in chemotherapy. Cisplatin brings about programmed cell death or apoptosis in cancer cells by damaging the cell's genomic or mitochondrial DNA and blocking its replication and repair. However, cisplatin has side effects caused by overdose and, if taken in excess, accumulates in the kidney and causes toxicity. Another problem is that sometimes cancer cells develop resistance to cisplatin therapy, resulting in a relapse of the disease. But don't worry, doctors are now using combination therapy, where 2 or more different chemicals which act differently on cancer cells are used along with cisplatin to reduce the side effects of cisplatin and overcome the problem of resistance. Plus, now with the advent of nanodrugs, cisplatin drugs can be delivered in the form of nanoparticles directly to the target cells, thus reducing their toxic effects. What are you doing while listening to this podcast? Whether it's driving or cooking, your brain has remembered these skills because of continuous practice. How does this work? The brain is made up of specialized cells called neurons. These neurons communicate with each other through a junction called the synapse. The presynaptic neuron transmits the signal to the receptors of the postsynaptic neuron. Every time we practice a particular skill, a specific pathway of neurons in the brain is activated over and over again, and thus the synapse is strengthened. In the strengthening process, the number of receptors in the postsynaptic neuron goes up, making the process a whole lot faster. However, if the synapses are strengthened only due to continuous stimulation, the target neurons start to fire too much, and their ability to change reduces, which in turn prevents the formation of new memories. This is called runaway strengthening. Similarly, synapses that were never used will deteriorate and then can't be used either. Since there obviously isn't a storage limitation on the brain, there must be a process to ensure this doesn't happen. In a recent study, researchers from the Indian Institute of Science Education and Research, Pune, have shown that the release of calcium from the neurons in specific synapses promote their reuse. The receptors in a synapse rely on calcium from the outside environment for signaling, and it was found that strengthened synapses had an additional organelle called the endoplasmic reticulum, or the ER, in the postsynaptic neuron. Now, the ER is involved in calcium storage, and I just told you that synapse regulation relies on calcium. So the researchers found that the ER tunes the amount of calcium released at the synapse and thus regulates changes in synaptic strength. As a result, the ER provides a braking mechanism to decrease runaway strengthening. Recent studies have shown that some mutations linked with a higher risk for Alzheimer's disease occur in genes that code for proteins involved in the ER's calcium regulation. Thus, understanding the ER's role in long-term memory can help understanding neurodegenerative diseases. Speaking of neurodegenerative disorders, to date, we know very little about these conditions and a possible cure is still a far cry. But a recent study by researchers from the Ramya Research Institute, Bengaluru, in collaboration with researchers from France and Chile on the biomechanics of axonal atrophy may take us a step closer to a cure. Let me just slow this down. The tail of the neuron is called the axon. Swellings along the axon is known as axonal beading and are commonly found in the neurons of people suffering from neurodegenerative conditions. Besides beading, normal axons also undergo a process of retraction during axonal rewiring. This process assists in the creation of new neural pathways to store information. However, in the case of diseases, this retraction causes the membrane of the axons to permanently recede from the axonal region and results in neuronal loss. The researchers knew that cytoskeletal filaments of the neuron, which are responsible for its structure, is disrupted during disease and are thus accountable for the process of axonal beading and retraction. By using cultured neurons isolated from 8-day chick embryos, they used 3 different drugs that uniquely disrupted the cytoskeletal elements to determine how neurons reacted when this happened. They were thus able to determine the biophysical processes behind axonal beading and retraction. Given that nearly 4 million people in India are currently affected by dementia, the study promises broader strategies for cure by revealing the mechanical factors involved in neurodegenerative disorders. Thank you for listening to this episode of Research Matters podcast. See you next weekend. For more such podcasts and news, please log on to www.researchmatters.in.